Bismillahirrahmanirrahim. Now these are some questions on inheritance, which is my video for helping you on the exam on Tuesday, for you to handle any question which is on inheritance. So if you just go through these, maybe they'll become easier for you as you revise this for the final time. Now this is a question which is very interesting because it has mixed up two things. Number one, it's got some definitions, and then it's given you a question on genetics as well. Catalase is an enzyme that breaks down hydrogen peroxide inside cells. Red blood cells contain catalase. So it's an enzyme ASE. Some dogs have an inherited condition in which catalase is not produced. This condition is known as acatalasia and is caused by a mutation in the gene for catalase. Define the term gene and gene mutation. Gene is a length of DNA that codes for a protein. Gene mutation is a change in the base sequence of DNA. So length of DNA that codes for a protein, that means if this is a chromosome and this is the portion of DNA, so this is the gene for eye color or gene for skin color or gene for hair color, then what happens in a mutation? There's a change in the base sequence. The base sequence is ATGC AT. Now if this becomes AT, so there is a change in the base sequence. First this was ATGC AT, now it is AT. So this is the bases. You see, you have four bases: adenine, thymine, guanine, and cytosine. And A always pairs with T, and G always pairs with C. So if this is the normal one, this is the mutated one. So mutation is a change in the base sequence of the DNA. B part of the question: a geneticist was asked to investigate the inheritance of acatalasia in dogs. The normal allele is represented by big B, and the mutant allele is represented by small b. The geneticist made the diagram in figure 3.1 to show the inheritance of acatalasia in a family of dogs. The shaded symbol indicates the dogs with acatalasia. So acatalasia happens when the person is big B, small b, when the dog, is big B, small b, because it's a recessive disorder. That was in the question. It said that the inheritance and the shaded symbols indicates. So that means this one is big, small b. This is small b. So first you write out what is the recessive trait, and that is, you've got to get it from the question. What was the recessive trait? So this is going to be small b. 2 is small b. Now when you've done this, then the whole question becomes very easy. You see, what you have to understand is now the other ones, the other people who are the normal ones, this is the normal one and this is the normal one. These either have to be this or they have to be this. Can't be anything else. So it is very easy to figure this out, because you have to understand is one is the normal people. So either this or this. But why can it not be this? It has to be this. And this also has to be this. Why? Because a small b has come from here and a small b has come from here. So that is why this is small b. This is shaded. So this is a small b which has come from here, and this small b has come from parent 1, and this parent has children 4 and this one. So they've asked you state the genotypes of the dogs identified as 1, 2, and 3. So 1 is big B, small b, 2 is small b, and 3 has to be what? Becomes very simple, because it has to be big B, small b. Why? Because one small b has come from this parent and one small b has come from this parent. You have to get one small b. In order to have a child with small b, small letter, small b, then you have to have one small b coming from the male parent and 3 is the female parent. It's a circle. Circle shows a female parent and square shows a male parent. And then of course similarly in the children, then of course we have that. So parent 2 and 3 have 2 children, 5 and this one here. This one is then married to this one, and they have this child. So you must be able to read genetic diagrams, right? So 1 is big B, small b, 2 is small b, and 3 is also big B, small b. Right? I hope everybody's figured this out. Geneticists crossed dog 4 with dog 5. Approximately half of the offspring had achalasia and half the offspring did not have achalasia. Complete the genetic diagram to show how this is possible. Okay, let's look at 4 and 5. So 5 is this homozygous recessive. And what is 4? Okay, 4 is normal. So now you can see 4 can be either this or can be this. But then it says that approximately half of the offsprings had achalasia and half did not have achalasia. So you see, if you have this, then all will be this. These are all normal. But it said half did not have and half of the offspring did have. So that is why this cannot be this. It has to be, then it becomes easy. So you can do a Punnett square here as well. And a Punnett square is very easy to do. You just have to write the male gamete you could just write it like this, or you could make a Punnett square and do it like that as well. So anything would be correct because a Punnett square is actually, this is what it's depicting. Now, anything else they've asked you? So offspring genotypes and offspring phenotypes. So you're given all that. State the name given to the type of cross that you have completed, and that's a test cross. A test cross is when the dominant one you don't know. When you don't know it is this or this. And then you cross it with a known recessive. Then you know if it is this one, then all will be the capital, the dominant trait. But if it is this one, then some will have the dominant trait and some will have the recessive trait. So this is what is called a test cross. I'm sure you're going to get a question on test cross. Question number 2. Anthocyanin is a red pigment found in carnation flowers. Some carnation plants have a gene for making anthocyanin. A flower grows red carnations. Describe how growers selectively breed plants. So you cross the plants or you breed the plants with a desired feature. You choose the offsprings for the desired features. Then you cross the plants showing the features with the original variety. Then you repeat this over many generations. And then you transfer the pollen and then you cover it up with a bag. And then you wait for the seed formation and then you collect the seeds and then you plant the seeds. So how do you word it? Cross parent plants with desired features, choose offspring for the desired features, cross plants showing features with original plants, and you repeat this over many generations of crossing and selecting, and then you bagging the flowers after transfer of pollen. Bag means that you cover the flower and you transfer the pollen, so they don't want any other pollen to contaminate your experiment. Then coming to the part 2, explain the advantages of using sexual reproduction to breed red carnations. So there are two parents are required. Offsprings might not all be red. It's very time consuming. So this is one of the disadvantages of sexual reproduction because you see when you cross them, there's a variation which can be found. So two parents are required. Variation in offspring might not all be red. It's very time consuming. Then harvesting the seeds and finding pollinators can be difficult, or it's wasteful in the content of unused pollen. So, disadvantages of using sexual reproduction is two parents are required, so gametes are required. The variation in offsprings might not all be red, or it's very time consuming, and harvesting seeds and finding pollinators can be difficult. Then we come on to B part. Meiosis is necessary for sexual reproduction of carnation plants. Define the term meiosis. What is it? It's reduction division. It's reduction division, and there are only 2 marks. The other is chromosome number is halved, diploid to haploid, results in genetically different gametes. So reduction division, chromosome number is halved, diploid to haploid, results in genetically different gametes. Carnation plants show codominance for the anthocyanin gene. There are two alleles. F raised to the power A allele for anthocyanin pigment, which is red flowers, and Fn for no cyanin pigment, which gives white flowers. State the genotype. State the genotype of a carnation plant that is heterozygous. So it has to be heterozygous, means Fa and Fn. Heterozygous. Now describe. Now they told you it is codominance. So one is red and one is white. Describe the phenotype of a heterozygous carnation plant for this. Whenever we use two letters, something raised to the power something, then it is for codominance. So this is going to be red into white, is going to be some shade of pink. So pink flowers. Then it says, but this was sort of, you could get it from the question, codominance. Codominance means both show their effect. And as I say, whenever we use something like F raised to the power A, so this is a phenomenon of codominance. We didn't say capital F and small f, that's it. Then it's dominant and recessive. But here we said something F raised to the power A and F raised to the power N. This is given in the question. So whenever there is such things used, then it's codominance. So red into white would be something pinkish sort of a shade. Then it says that breeder crossed Fn carnation with Fan plant predict using another proportion of pure breeding carnation plants in the offspring. So that was easy to do. You had to write the gametes, then you had to do the Punnett square. You could do that. You could write it like this. Offspring genotypes were these, red and pink, and the proportion of pure breeding. Pure breeding means when they both homozygous dominant is also pure breeding. Like if I use this, like this is pure breeding, this is also pure breeding. Now in this situation, this was pure breeding. This is heterozygous. So heterozygous is not pure breeding. That's another way of saying it. Pure breeding means either homozygous dominant or homozygous recessive. They're both pure breeding. Sex in cats is determined by the same way as in humans. Complete the diagram below to show how sex is determined in cats, male into female. And then, of course, they draw the Punnett square. So male would be XY. So there would be an X here and a Y here. And then this would be XX. This would be XX. This would be an XY. And this would be an XY. So offspring ratio is 1:1. Or you can say 50% male, 50% female, or you can say 2:2. A scientist investigated the inheritance of fur color in cats. The gene for coat color is recorded on the X chromosome. The gene has two alleles, B for black and small b for orange. The X chromosome with the allele for black is represented by X raised to the power B, and the X chromosome with the allele for orange is X raised to the power small b. A female cat can be a mixture of these colors described as calico. Figure 3.1 shows the inheritance of this condition in a family of cats. So orange male, black male, and all this story. And then let's look at the question on it. State the genotypes of cats 1, 4, and 5 in figure 3.1. 1, 4, and 5. Now you can see that 1 is the orange male. So the orange male has to be what? It says the X chromosome with the allele for orange is X raised to the power small b. So 1 has to be X raised to the power small b and Y. Y does not have anything on it. The reason is, you see, this is the X chromosome, this is the Y chromosome. So the B is somewhere here. So there's nothing here. So Y does not have anything on it. That is the question. And when we are doing a sex-linked disorder, they've given you this question knowing that you know what is the sex-linked disorders. So X raised to the power small b Y, that will be number 1. Then what will be 4? What is 4? 4 is a black male. So black male would be this. So it would be X, big B, and Y, nothing on the Y. And then cat 5 is a black female. So cat 5 can only be X big B and X big B. Why? Because it was given in the question that big B is black. This would be the genotype, so this is how you would write it. Then coming to the last part of this question, coat color in cats is an example of discontinuous variation. Explain why coat color is an example of discontinuous. So direct question, they are distinct phenotypes or they are distinct categories. No range of color, there's no shades, different 10 or 15 different shades of color as controlled by genes. And plus this is not affected by the environment. Like we see the height of stems, the length of stems is affected by the environment if they didn't get enough light. So that is a continuous. You see, what you have to understand, continuous variation is affected by environment. Discontinuous variation is not affected by environment. For instance, a person has blood group O, and even if you don't give him enough proteins or you don't give him a good diet, it doesn't have enough carbohydrates in his diet, he will still remain blood group O. Discontinuous variation is not affected by the environment. So distinct phenotypes, no range of colors, so it can't be continuous variation, it's controlled by genes and is not affected by the environment. Question 4, figure 5.1 shows the nematode, Kano elegans, whatever, it's a long, difficult name. State the genus. The genus is always this one. Genus and then species. So you just have to write that again. So you just wrote that, and that is the genus. State two structural features of nematodes. What is that? They're thread-like bodies and they have unsegmented body. So, I mean, this is of course part of 0610 syllabus. Thread-like bodies, unsegmented body, hydrostatic skeleton, body tapers pointed at both ends, and mouth and anus, through mouth and anus, relatively large pharynx or sucking mouth parts. Nematodes feed on dead and decaying material. Explain why this gives nematodes an important role in ecosystems. Why is it so important if they are work on dead and decaying? Yes, very prevents accumulation of dead matter. Otherwise, people would be just buried in the dead if nobody decomposed. If trees fell down, were not decomposed, elephants died, not decomposed, anything dying would not decompose, or they would be lying around, and we would be buried in the dead. So prevents accumulation of dead matter, recycles nutrients, releases carbon dioxide. All these material, when it's decomposed and the bacteria and fungi respire, and carbon dioxide released back into the atmosphere, and that carbon dioxide is used for photosynthesis. Also, it decreases particle size of food for decomposers. And this allows the energy flow in the food chains and the food webs and the ecosystem. So removes organic waste, recycles nutrients, releases carbon dioxide because bacteria and fungi respire and release the carbon dioxide. And this carbon dioxide is going to be used for photosynthesis, and also it results in the energy flow in the food chains. Now figure 5.2 shows the life cycle. The diploid number of this species is 12. Adult male, which has both male and female sex organs. Diploid body cells, P, sperm, egg, Q. Embryo passes out of adult and continues development, not drawn to scale. Suggest why there is very little genetic variation in the offspring of the adult nematode shown in figure 5.2. Gametes are from the same individual, self-fertilization. Only new source of variation will be a mutation, and variation is usually produced by meiosis. But here, what do we have? We have the diploid body cells of the adult, and they are resulting in the formation of the sperm and the egg. Adult, which has both male and female sex organs. So this is why there is not going to be very little genetic variation. So gametes from same individual, self-fertilization, only new source of variation is mutation. Variation is produced by meiosis. State the haploid number. It was 12, so diploid was 12, so haploid will be 6. Now part 3 says explain why meiosis occurs at P and mitosis occurs at Q. So meiosis at P here, we're talking of this situation, right? Now at P, why is meiosis? Because it's reduction division. Chromosome number is being halved, prevents doubling of the chromosome number with each generation. And gamete cells are always haploid. And gametes or the sex cell production is taking place because the sperms and the eggs are being formed. So the sperms and the eggs are always haploid. While at Q, then this cell, the zygote has been formed. Now the zygote is going to divide by mitosis, so that now we can have growth, two cells, and then four, and then six, and then eight, and then a ball of cells, and then it's going to form an organism. So growth is taking place, producing genetically identical cells or more diploid cells, and then of course the body of the organism will develop. So meiosis, reduction division, prevents doubling of chromosomes at fertilization. You see, if the gametes weren't haploid, then every generation the chromosome number would double. And gametes are always haploid. Mitosis at Q, growth is taking place, producing genetically identical cells and more diploid cells. C. elegans was one of the first organisms to have its genome sequenced. An organism's genome is the sum of all the genetic material. Gene sequencing identifies all the component parts of the DNA that makes up the genome. State where DNA is located in a cell. It's on the chromosome. The chromosome is made up of DNA. Or you could have said that, or you could have said in the nucleus, and also DNA is found in the mitochondrion. So in the mitochondrion as well, DNA is found. Mitochondria has its own DNA. So DNA is located in a cell, and if you say bacteria as a cell, then also in the plasmids. So you could have said anything for 2 marks. My chromosome, nucleus, mitochondria, plasmids is also been allowed.